Hello, good afternoon, and welcome everyone, and thank you for joining our webinar. Happy spring to all of you. This afternoon we're going to speak about Capricor Therapeutics. I think that all of us are quite familiar with the HOPE-1 study and with Linda Marband, who is the founder, president, and CEO of Capricor. So today Linda's going to update you on Capricor's study and where they're going and what they hope to achieve and how we can think about this going into the future. So with that, Linda, I'm going to turn this over to you.
Thank you so very much, Pat. Good morning, good afternoon, and good evening, wherever you are. Thank you so much for joining us. And shown here is just a standard forward-looking statement slide. That means that Capricor is a publicly traded company, and you can find information about us in various places in the SEC website. And I think most of you, as Pat said, are familiar with Capricor, with HOPE-1 and the promise of that study, which was recently published in the journal Neurology, and then the ongoing HOPE-2 clinical trial. However, there's been a lot of things going on, as often happens in the clinical trial arena, and so we thought that what we would do today, thanks to PPMD, is provide you with an update on where we are, where we anticipate we are going, and then take any questions from you in the community so that any misconceptions can be corrected. Right here is just a very simple agenda. I'm going to briefly go through the original trial design and the endpoints that were selected. Then I'm going to give a momentary update on our meeting with the Food and Drug Administration, where we had a Regenerative Medicine Advanced Therapy meeting. Regenerative Medicine Advanced Therapy designation, or RMAT, as it's commonly called, is like breakthrough for cell and gene therapies. So it allows pretty specific and directed assistance from the Food and Drug Administration, the FDA, as you get your product towards commercialization. We had a safety issue with the clinical trial that I'm going to talk a little bit about, and what we did to hopefully put an action plan in place to prevent that or attenuate that from happening in the future. I'm going to go through very briefly, and I emphasize briefly, this is not a medical update call, but really a corporate update call, what the pre-medication regimen is and what we put in place. And then I'm going to talk a little bit about our plans for going forward, and finally I'll conclude with questions from you, which I hope will provide more clarity beyond what I'll already provide. So shown here on the slide is really just a refresher for those of you that have been following for a while, those of you who haven't. We originally planned the HOPE-2 clinical trial based on the results from the HOPE-1 clinical trial. HOPE-2 is a randomized phase 2 double-blind placebo-controlled trial. The goal of the trial was to evaluate, of course, safety, but primarily focusing on efficacy of IV delivery of our cell therapy product called CAP-1002, which was going to be given over the course of 1 year 4 times to each patient. So once a quarter, a patient would come in for an infusion of 150 million cells. We were anticipating up to 15 sites in the United States. Craig McDonald at UC Davis is the national PI. And what we were going to be doing is following these patients. The patients would be later stage Duchenne muscular dystrophy patients with a decreased upper limb strength. That would be one of the most important inclusion criteria. A stable steroid regimen, and cardiac and respiratory function to the point at which they were not suffering from significant cardiac or respiratory impairment that would attenuate their ability to perform in the clinical trial. And we're going to be evaluating a variety of secondary and exploratory endpoints, but the primary efficacy endpoint was going to be the performance of the upper limb, the mid-level. And that's because what we were trying to do is evaluate whether we could recapitulate the results that we saw in HOPE-1, where patients did see an improvement in their ability to move their hands, arms, and elbows in response to the cell therapy, or what we believe was the response to the cell therapy. And so that was the goal of the clinical trial. So what's happened since that time? And that's where we're going to go now and talk a little bit about our journey forward. So in December of 2017, we had the RMAT meeting with the Food and Drug Administration. We had several advisors and observers attend the meeting with us. Among those was Pat, so she could provide the most clarity to the community as to what the FDA was saying to Capricor. Craig McDonald is our PI. Michelle Eagle from Adam in the UK, where the PUL was developed and first tested, and then Lee Sweeney from the University of Florida, who has world expertise in muscle physiology, strength, and measures, particularly, although not exclusively, in Duchenne. What the FDA talked to us about, and was a really very productive and collaborative meeting, was that they liked the idea that we were attempting to provide therapeutics for the later stage patients, those that are either at the edge of losing ambulation or have lost ambulation, and focusing on something for those patients to help preserve upper limb strength. They felt that the PUL 2.0, the newer version, would be a better measure than the 1.2. There's not a lot of difference in the two measures, and I can answer questions on that if you have them. But what I can say is that the metric has become tighter. The reason that the FDA likes the 2.0 as a primary efficacy endpoint versus the 1.2 is really because they feel that future validation with 2.0 will be better. So while 1.2 is validated, they like the fact that 2.0 will be the gold standard moving forward, and they want to sort of put everybody on the same playing field. I think the community, our investigators, and the FDA are very excited about this particular clinical trial, the HOPE-2 one, not only because of its potential opportunity to help people, but also to help provide information on this very important measure for patients that even just a few years ago, once they became non-ambulant, were, so to speak, forgotten by the medical community, and that can no longer happen without there being some type of a response.
So, Linda, I'm going to just interrupt you for 1 second and say, the PUL is the performance of the upper limb. So can you just briefly explain what the PUL is, what it's doing, and what the change in from 1.2 to 2.0, so we're all sort of on the same playing field during the discussion?
Absolutely, Pat, and I'm very sorry. I think I work in this vernacular all day, every day, so I forget that everybody doesn't. So
yes, no, it's no worries.
Is the performance of the upper limb. It was a measure that was developed by patients, families, physical therapists, and physicians that allows the evaluation of the strength and the upper body of patients, and that could be anybody, but in this situation, Duchenne patients. So this starts with the entry criteria is the shoulder. Can you raise your arms above your head as an entry criteria that starts to look at whether you still have mobility in your shoulder and your torso. And then that's called the shoulder area. For the mid-level PUL, which is what we have focused on, that's really focusing on almost exclusively elbow, bicep, forearm, so things like being able to drink a glass of water or comb your hair or brush your teeth would be measures that would be measured by the mid-level PUL. And then the distal is really just fine motor control. This would be hand motion. So ripping and tearing paper might be one of the measures, being able to hold a pen and write, all the things that you would do with the fine motor control. Now, what's really interesting about the performance of the upper limb as a clinical indicator is that the shoulder area is lost quickly, usually when patients begin to lose their torso muscles, and then usually soon after ambulation is lost. The shoulder can decline very quickly. So that's not always the best measure to track in a clinical trial because it can be sort of one and done. You lose it and then it's kind of not measurable. The distal, by contrast, the hands are the last to go. So they are very slow to go. They are sometimes years before you see a change in a patient's ability. But the mid-level is actually just sort of that Goldilocks sweet spot where within a year you typically would see changes in patients that have lost ambulation or close to and then have also lost their shoulder mobility. So it's a very tightly corroborated measure with tasks built. There's 21 tasks that are built in, all to look at functions that would be similar to activities of daily living. So, for instance, I talked about drinking a glass of water or, moving a joystick on a wheelchair. Those are things that you can't actually measure, but what you can do is have patients lift and move cans or move their hands around on a table so that you can see the mobility that they have. Does that sort of provide a little bit of clarity, Pat?
Yes, thank you so much.
Of course. So what the FDA said at the end of the meeting is that they felt that we were on the right track, that they would review the end-of-phase data to determine registration potential, but felt that if we provided clinically meaningful evidence of changes in the performance of the upper limb, as well as other measures of efficacy that would be corroborative of performance in skeletal muscle, that they would consider that for registration. We also realized as we went from POL 1.2 as the primary efficacy endpoint to 2.0 that we could potentially reduce the sample size, and I'll talk about that in the next slide. So, whoops. Let me just go back. Okay. So, we realized that we could reduce the sample size from 84 patients. We anticipated that based on power analyses, we could go to between 40 and 50 patients. We were going to do the exact same trial design, follow the patients for 1 year, where we did 4 infusions and look for changes in efficacy at the end of that time point. What happened along the way that changed this course just a little bit is that we had an allergic reaction in 2 patients that would be quoted as anaphylaxis. These can be life-threatening events. Thank goodness both patients, one from our HOPE open label extension group, that was a patient from HOPE 1 who was a standard of care treatment patient who came in to get cells, and then the other was a randomized patient from HOPE 2. Both had a very similar response in 2 different locations where they developed symptoms of anaphylaxis, being unable to breathe, tightness in their chest, fast heart rate, symptoms of severe allergy. Thank goodness the medical teams were right on it. The young men were fine. They went home the next day with a scary story to tell their friends and family. But as a result of that, we went on a voluntary dosing hold. We didn't want to put patients at any added risk than we have to when we put them through any types of medical regimens. And we looked into the causes of the allergic reaction. I will tell you, sort of in a preview to the end of the story, we still don't have an idea what caused the allergic response. The patients have undergone assessment for what they might be allergic to, and we still are trying to figure it out. We don't have any indicators. But what we found out while we started talking tremendously and quickly to thought leaders in the immunology and in the allergy world was that this type of a response is not uncommon when you treat with a biologic. And so therefore there's a variety of pre-medication regimens that are out there, and I'll talk about that in the next slide to attenuate these types of responses. We also, based on the FDA, as I mentioned, switched to the performance of the upper limb 2.0 as the primary efficacy endpoint. This is really almost just a paper change because we were measuring it anyway, so it doesn't have much implications except that it allowed us to recalculate the trial size, and so we would be able to use approximately 40 patients in a randomized double-blind placebo-controlled trial and still potentially achieve indications of efficacy with the appropriate power. The most important thing is that we decided, based on the time that we lost while we were on the voluntary dosing hold, which I can say thanks to my team, who were terrific in tracking down pre-medication regimens, and our leaders, Craig McDonald at UC Davis primarily, we were down for a very short time. So we went on voluntary dosing hold December 21st, and we resumed treatment in February of 2019. So we weren't down for very long. But as a result of the time that we were down, plus capital needs of the company, which I'll talk about in a moment, we decided to do an interim analysis, and I'm going to talk about that also in the next few slides. So first, and I think many of you are wondering, what is a pre-medication regimen? or pre-treatment regimen. Pre-treatment regimen is something that you do. You give medication prior to, for instance, infusion of the therapeutic, in this case it would be CAP 1002, to blunt what might be an allergic reaction. So the safest way to do this is you say all patients get it. You don't know who's going to mount an allergic response until they're doing it, and so in order to be safe rather than sorry, all patients receive this pre-infusion treatment regimen, which is actually quite simple. It's a sort of a polypharmacy of high dose of steroids, antihistamines that are taken orally at home before they come into the center, and then they get a bolus infusions in the center before they get their cells. We haven't heard that anybody has had any side effects from the medications. They're pretty common medications like Benadryl and ranitidine, and then they also get steroids. And as I mentioned, we developed our pre-medication regimen, and we adopted the one from UC Davis based on doing a significant amount of research within the literature, but also talking to thought leaders, people that are allergists, immunologists, pediatric endocrinologists, and intensivists. So we felt pretty comfortable with the regimen that we came up with, and the good news is it's very similar to a lot of others that are out there, so we know that there's nothing about it that makes it unusual. To that end, and here's the pre-infusion guidance that was actually approved by our steering committee and the Food and Drug Administration, and that basically we recommend this pre-medication regimen, which is, as I just described for you. But if a center, if a medical institution has one that they have been using for a long time or are more comfortable with that has a similar components, they're at will to use that. So the principal investigator, the physician at each site, gets to make that decision. As I mentioned, we've had over 20 infusions since we started this regimen, and we haven't had any issues with it at all, except one patient had some rapid heart rates a few weeks ago, and we didn't think that this was potentially related to this situation, but he also went home and is doing fine, and so we feel that we've managed the problem effectively at this point. So based on a lot of the information that's circulating around, we've had some safety events, we've had some issues with raising funds through the company. There's a lot of discussion as to whether, HOPE-2 is a trial that should be done. And we at Capricor, but also in the Duchenne community, we hear a lot of people saying HOPE-2 is an important trial. It needs to get done. It's one of the only therapeutics that is out there that is directly addressing inflammation and fibrosis. So if the data in patients mirrors the data that we got in the mice, we should really see something that not only knocks down the inflammation, which, as you know, is one of the most egregious aspects of the disease process, and then also addresses the fibrosis, which as a result of the inflammation is the death of the tissue in and of itself. We saw really terrific improvements in the MDX mouse, and what we have, and has not been published but is exciting new data, is that the exosomes, which is the active pharmaceutical ingredient, or the little bubbles that are released by the cells that carry the message of healing and regeneration, are actually taken up by the satellite cells. And that's something that is of prime importance in potentially having a long-term effect in Duchenne muscular dystrophy. The HOPE-1 clinical trial, we showed clinically relevant improvements in the performance of the upper limb and cardiac function and structure. That paper was published in the journal Neurology in January of 2019. So we feel that, we've had peer review of this data, and it looks like there's really something there that could be taken all the way through if the data bears fruit. And then finally, as I mentioned earlier, the FDA is very supportive of the program. They like the therapeutic. For the most part, it's safe and would be something that could be adopted by the community. And also, and what I think is one of the most important things, it's one of the few therapeutics that are targeting the later stage older DMD patients. And these are patients that, the gene therapies that are out there that are looking so promising may not work as well for, at least in iteration 1, because of the amount of inflammation and fibrosis. So our goal at Capricor would be to provide a therapeutic that could attenuate the inflammation and the fibrosis, but also provide the way or pave the way for gene therapies for the future.
Well, I think one other thing back on that slide, Linda, is important to note is that the gene therapies, one of the concerns about gene therapy is that it doesn't get into the satellite cells. So as this potential therapy moves forward, should it be approved, it would be really terrific to preserve those satellite cells and as gene therapies improve over time.
Exactly. Thank you, Pat. So what we've finally decided to do after, thinking a lot about this and really paying attention to why we think this clinical trial should be done, what we thought is that we could do an interim analysis. So what is the interim analysis? Why are we doing it? And how it's being laid out? And an interim analysis allows you to look at data, and there's varieties of ways these can be done. They can be done sort of the most non-invasive, which would be an evaluation of futility. Is this looking like this one and done, not working at all? And the data is only seen, for instance, by the DSMB, the committee that reviews data and makes sure that patient safety and concerns are always front and center. And so the companies or the patients or the community even has no idea what those results are. The DSMB just says, yes, you can continue. That is the least invasive all the way to what we consider the most open or invasive, which is actually what Capricor decided to do, which is you fully unblind, you look at all the data you have to this date, and you see whether you have a biologic signal. And if you do, then obviously the path forward becomes more clear. The goal then would be to finish enrolling the trial, get the data, and potentially register the therapeutic. If the data doesn't look promising or it looks like it's not working in the way that we had hoped or thought, then it allows sort of reconfiguring of a program without putting more patients, more families, more physicians, and frankly more of the community into a position where they are adjusting their schedules and their lives to go get this therapeutic. So what we decided to do is look at the data when all of the patients who received their first infusion prior to the safety event in December, so anybody who got their first infusion prior to that event, hit 6 months, which is approximately in June of this year. We'll look at the data from all those patients, looking primarily at the performance of the upper limb, some of the other measures of skeletal muscle function, because remember I told you at the beginning that the FDA wants to see corroboration between the endpoints of skeletal muscle function and improvement, as well as the performance of the upper limb. We'll look at cardiac structure and function, which is where we started out in the world, thinking that we would have a therapeutic that would treat the heart, so we're still majorly focusing on that in our internal programs. And if we see something, then the goal would be to go out and raise the capital to complete the trial and then continue on the path forward. And it's the most effective way of getting that information, both for ourselves to determine our path forward, for the families that are most interested in treating their children, and then the investment community so that we can raise the funds to do this. So, in conclusion, I guess what I'd like to say is what should patients and families do now? Please, if you are already in this trial, please complete all visits as required because we're doing the interim analysis on relatively few patients. There's less than 20. We really need everybody to get in there and get their visits so that we have all the data points that we possibly can have. If you want to be in this trial, please stay tuned to see the results of the interim analysis, and if the signal is there and we reopen the enrollment, we'll be very excited to have you come in at one of the many sites that we'll have up and running. And then the other issue I wanted to address, because I know it's been in front and center of your minds, as it should be as parents and family members and caregivers of children with Duchenne, is there a safety issue with CAP-1002? And the answer is we don't believe so. the pre-medication regimen seems to be working, and there don't appear to be any other safety issues that are impacting the conduction of this clinical trial at this time. So we think that it should be relatively safe, and we would encourage you to stay forward with your therapeutic. And then finally, we would like to open the line for any questions that you have.
Yeah, thanks very much, Linda. That was really, really helpful. So a couple of questions have come in, Linda, and one of them is really about should you see a trend, would you expect then this drug could be applicable to children who are ambulatory to reduce inflammation and fibrosis, preserve ambulation? So would you see a generalized benefit that could be expanded to other populations? And then should you see a trend, will you consider opening studies in Europe as well?
Yeah, so the answer to the first question is absolutely. You know, we never developed these cells with the idea that they would stay exclusively focused on the later stage patients. It was just that the metrics of measurement and was most beneficial because we started off treating the heart, and so these were later stage patients. So it was sort of chance favoring the prepared mind that we were initially targeting the later stage patients, plus there's very little out there for them. But then we would, of course, want to expand to the younger patients as well, so that we can preserve muscle as well as treat damaged muscle. And then in terms of Europe, you know, the goal, of course, would be to be worldwide at some point. We're focusing right now on the U.S., and then when the data bears fruit, we'll decide a path towards expanding worldwide.
Good, thank you. And I had a question in one of your very early slides where you talked about you're delivering 150 million cells. How did you get to the dose of 150 million cells? And these, you're delivering the same dose consistently across, right?
Yeah, so it's 4 doses of 150 million cells, and we did that by doing some dose response studies in animals, both small animals, the MDX mouse for efficacy, and then typically what we do is do toxicology or dosing studies in large animals looking for beneficial effects primarily in the heart, which is how we did it. And so that was the sweet spot where we saw the best effect without any toxic results.
Okay, great. Thank you. That's very helpful. And a couple other questions is, you know, obviously in quarter 3 you're going to have the data that will from the interim analysis and make some decisions around that. So how will the community learn about that? Will you come back and do a webinar? Will you publish something first? So if you can give us a little bit of the timeline of how will we know when you know?
Yeah. So as you know, Pat, one of my goals has always been to maintain close contact with the community. So our general plan is that when we get the top-line data, of course, we'll analyze it. I'm saying sometime in Q3, as everybody knows, it takes a while to get the data in and downloaded and cleaned and all the stuff that they have to do with data. And then as soon as we have the data, whether it's a go or a no-go, I'll reach out and we'll schedule another one of these webinars where I will walk the community through our findings and then our plan to go forward. Now, I want to be clear while we're talking on this issue, until we raise the funds, we can't continue the trial. We would have to go back out and raise funds. So we couldn't start treating patients right away if we saw positive trends, but we certainly believe that it would provide enough energy in the investment community to support it financially, and then we would be able to then start enrolling patients soon after that.
Right, but at this moment, Linda, the company has sufficient funding to continue to the interim analysis so that you can have an understanding of whether this is a go or no-go and what the learnings are.
Absolutely, and that was why we decided to do the interim analysis when we did, because we have more than ample funding to get through that and to data decision.
Yeah, and you talked about if you don't see a trend here, you're going to sort of go back to the bench and take a look, because I think exosomes as a potential delivery vehicle and as a potential for decreasing inflammation and fibrosis are really sort of that research is evolving at this moment, and we're hearing more and more about it. So can you, I know you can't say to me, here's what the plan is, but can you shed a little light on how you're thinking about that? Because anyway, even with a positive trend, you're still looking at exosomes as delivery vehicles. So can you give a little information around that research and where you're going with it?
Yeah, so the exosomes are very exciting. Anybody that works with me or knows me knows that I think that they are the wave of the future for biotechnology. Exosomes are tiny little bubbles. We call them nanometer-sized lipid bilayer vesicles, and for those of you that are not scientists, what that means is that they are a million times smaller than a millimeter, and I think most people have sort of a kind of a picture of a millimeter in their head. They're incredibly small. When they were first discovered in the 90s, they were thought to be the trash cans of the cell, because cells spit them out all the time. And so scientists just thought that they had junk in there that the cell wanted to get rid of. But then in the early 2000s, somebody very smartly opened them up and found that there were messenger RNAs inside, microRNAs, and RNAs are how proteins are made. So they are really cellular communicators. It's the Morse code or the communication language of cells. And so what we realized very quickly in the field is that the exosomes are how cells tell other cells what to do. It's how they give them directions, how they change behaviors, how we change our physiology. And so now the field of exosome therapeutics has exploded because we are trying to take advantage of their capacity as delivery vehicles to address all kinds of biologic problems, everything from cancer all the way through neuromuscular disease as diagnostics, you name it. We're also working on trauma programs with the U.S. Army. There's lots of different ways that exosomes can be used. And so, yes, what we're going to be doing, what I'd love to see happen is that we move away from the cells over the next, let's call it 5 to 10 years, and move into exosome therapeutics, where the exosomes, the active pharmaceutical ingredient, are actually what we'll be using to treat the boys and young men. And then we'll have, sort of a new class of therapeutics.
Right. So the cells do spit out the exosomes, but also exosomes are able to get into cells as well. So right. It's sort of this bidirectional communication that they do with these exosomes. Is that correct?
Yes, that's exactly right.
Good. Well, we are excited about this. And can you tell me, can you relate the size, because we're used to the description of the adeno-associated virus as gene therapies are evolving, and the size of the adeno-associated virus, and can you give us some hint about how that size is relevant or related to, what it looks like compared to the exosome?
Right. So, one of the things that the field is doing is thinking about putting AAV inside the exosome. So the exosomes can actually house the AAV and potentially use it to deliver the gene therapy. So we and others are also considering that as an opportunity. So the AAV is small enough to fit inside an exosome potentially. Exosomes can also hold more gene product, right? So what people are playing around with is, could the microdystrophins be made bigger so that they could, pack more into the exosomes, which is also a possibility. So they're bigger than a virus, but obviously small enough to get across the cell membrane.
Yes, thank you. So it is very exciting research, Linda, and we are really enthusiastic about it and certainly wish you very well in this study. I wanted to also say, in the HOPE-1 trial, when you delivered through the cardiac cath, I think it has to be known that cardiac cath had not been done in Duchenne muscular dystrophy prior to your HOPE study. And we were all a little, as you well know in our discussions with it, a little nervous about what happens to these kids getting cardiac cath, and will you see any side effects, and will there be any safety issues with regard to doing a cardiac cath? You've done that study successfully in terms of delivery of the cardiac cath and certainly the publication that was in neurology. And so we're really thankful for that, because this also opens the way to a gene therapy to the heart for these kids, because some of those adults just to rescue the heart. So in your proof of concept of being able to say that delivery via cardiac cath can be done safely to these children has really pioneered other research. So we thank you for all that you do. I certainly wish you well in this study. I hope you see trends, and I hope that someday that exosomes are delivering a bigger package than the microdisc that's currently being delivered, because I think we'd all in this community agree the bigger the dystrophin message you could give, the perhaps better out. Obviously, we don't have the data, but perhaps the better result. And so for your dedication and for all of us who are wishing for good drugs, better drugs, and certainly the safety issues around those. So I thank you for that, and I also thank you for the pre-treatment. I think that I'm probably going back and forth a little bit, but I think pre-treatment may occur with all of these biologicals. Gene therapy, they're doing pre-treatment, so this is not, again, an issue that has, it's new to Duchenne perhaps, but it certainly isn't new to the whole biological field. So I thank you for that as well.
Thank you. Yes, absolutely. Thank you, Pat. That was wonderful, and I really appreciate the support of PPMD, and certainly hope that the work that we're doing and the work that others are doing will ultimately make a difference in the lives of these boys and young men.
Yes, I thank you. And we have no further questions, Linda, so I really thank you for the update. We'll be anxiously awaiting that September update. We'll have our fingers crossed and appreciate the ongoing research with exosomes, but hopefully we'll see a trend in when you do the analysis, and then we can rapidly expand and hopefully deliver a therapy to everyone in fact, in this community who needs and deserves treatment. So thank you with that, and thank you all for joining us. This will be posted and online and available within a very short period of time, and Linda will come back to you if there are additional questions.
Thank you,
Pat. Thank you again for
Kaylin
joining us.
And the team. You guys are the best.
Thank you. Thank you for joining us.
Bye-bye.
We wish you well. Bye.
Bye-bye.